Hello, everyone, and welcome to our YouTube channel. We are Group 5 members of the Department of Cell Biology and Genetics, and today we'll be discussing on activation and collision as a mechanism of intermembrane trafficking. There are a number of ways by which lipid transfer exchange occur between the ER, Golgi, and mitochondria at membrane contact sites. Activation and collision is one of these mechanisms. So constantly, membrane-bound organelles and vesicles are constantly moving around in cells via motor proteins or cytoskeletal tracts, and this brings about close contact or a collision with each other. The activation and collision mechanism describes a reversible two-way pathway for lipid transfer during transient collisions between the donor and acceptor particles.
First of all, activation is basically the preparation step, which is meant to enhance the efficiency of lipid fusion between target membranes and the vesicles. And all in essence, the process of activation is just done to ensure the precise and controlled transfer of either biomolecules or neurotransmitters from the vesicle to the targeted site. Then there's collision, which occurs when the targeted membrane and the vesicle, they come in close proximity or they come in close contact, just to ensure the process of diffusion.
When membranes collide, proteins and lipids from one biolayer diffuse to the other through a process called lipid mixing. Now, this only occurs if both membranes are compatible. Certain proteins like SERTs regulate and catalyze this process of lipid mixing. SNAP proteins are molecular motors that drive the fusion of both membranes. We have B snares on vesicles and T snares on targets. Snare proteins on both membranes zipper together to bring them into closer position and promote lipid diffusion. Grab GTPase protein in their active GTP bound form on one membrane binds the effector protein on another membrane and joins them together. This increases the chances of collision and mixing.
Following collision and lipid mixing, the organelles of vesicle may fully fuse together to become one and may separate again in what is known as the kiss and run event. The activation and collision mechanism allows efficient transfer of membrane and content mixing between organelles without the need for vesicle building and fusion. It is important for processes like ER to Golgi transfer and also the nuclear envelope reformation during vitreosis. The high mobility of organelles in a cell increases the frequency of productive collision mechanism by which molecules can move between intracellular.
To conclude it all, activation and collision pathway was first described by Skett et al. 1987. Now, this experiment was aimed at explaining the kinetic similarity between activation and collision pathway and the diffusion mechanism. Now, this was designed in such a way that the red blood cell was labeled with cholesterol and was incubated with proteins we call the acceptor proteins, due to collision activated theory we've been describing since. Now, this was, according to the experiments, they found out that the environment of the experiments, that means the surrounding of the acceptor and the donor particles, affects the transfer of cholesterol from the red blood cell to these vesicles. Another thing that was found out in the experiments was that the environment, or let's say the transfer of the cholesterol from the donor to the acceptor particle is inverse to the size of that particle. Another thing is increasing the dilution of the concentration around both the donor and the acceptor particle also decreases the transfer of it. Now, there is a particular part, or let's say one of the acceptor particle that was used, which is the ghost particle. They call it the ghost particle. I can't really describe that much, but the ghost particle, they noticed that increase in the dilution led to zero transfer. Total, no transfer amount. So, activation and collision theory, or let's say the pathway, basically describe the activation mechanism of a particular lipid to be transported to a particular acceptor particle. And this is done by the mechanisms that were described by my mates, describing the activation process, which involves several processes of particles activated, and also collision theory, that is physiology, or let's say physical interaction between several membranes of different vesicles that are trying to interchange their internal material, and this is a very important mechanism in trafficking lipids across membranes. Thank you.